In this experiment, a monkey operates a robotic arm to feed itself marshmallow treats through a brain-machine interface. The monkey's own arms are restrained, and the movements of the prosthetic arm are controlled through the neural activity of the monkey. A multi-electrode array implanted in the monkey's motor cortex records the spike pattern of 116 single neurons. The movement intent signals generated by this population of neurons are decoded by a computer in real time and then executed by the robotic arm. Note that the experimenter randomly changes the location of the marshmallow, and the monkey is able to navigate the change in three-dimensional space, grab the marshmallow, and return it to its mouth. Remarkably, these actions are performed within a few seconds, similar to the use of a real arm, with approximately a 60% success rate. During the experiment, the activity of the 116 neurons is recorded and represented as a spike roster. Each row in the roster represents the spike activity of a single neuron over time. The preferred direction for each neuron can be determined based on its firing rate changes during certain kinds of movement. And in this dataset, the individual rows are grouped by the major tuning components, that is, the 4 dimensions of the experimental space, indicated by the color bars. Red for the x-axis, green for the y-axis, blue for the z-axis, and brown for the gripper, the 4th dimension in this setup. An algorithm calculating the population vector from the recorded neurons estimates the actual arm movement and controls the robotic arm. The graphs plot the movement of the robotic arm in millimeters along the x, y, and z axes. The state of the gripper arm is either open or closed, represented as a value of 1 for open and 0 for closed. The areas in darker yellow indicate the inter-trial intervals. Arrows indicate the moment the gripper closes on the target. Given the monkey's success in grabbing the marshmallow, it is clear that movement direction can be successfully predicted by the population activity of motor cortical neurons. The same axial datasets can be converted to a 3D representation to provide a more intuitive view of the motion and action of the robotic arm. The blue areas indicate when the gripper arm is closed. Note that the monkey opens the gripper before the marshmallow reaches its mouth. During the trials, the monkey learned that the marshmallow would stick to the gripper, so it didn't need to keep the gripper closed for the whole return trip. Success with these types of experiments in animals led to human clinical trials with paralysis patients. In a similar experimental setup, a human patient with tetraplegia has a multi-electrode array surgically implanted in his motor cortex. The firing of these neurons is then processed by an algorithm that allows the patient to operate a computer cursor in real time. At the beginning of the experimental process, the patient was asked to imagine moving the cursor and was provided with visual feedback that helped improve performance. After extensive training, as we can see here, the patient is able to perform a variety of tasks, such as moving the cursor to designated targets or opening a file in an email by imagining the movement. While this is remarkable, the levels of device control have yet to match the action of a human hand using a computer mouse, and the long-term stability of electrode arrays is a serious limitation. Significant work is still needed to create devices for long-term human use.